computer, and then I'm going to go back to my Blackboard and pull up our E3, and then we have the PowerPoint. Maybe we have the PowerPoint. There we go. All right, so just so we get this straight, so you're not confused, we're going to do gram-negative Coccobacillus. So we're going to start there, but then we're going to switch to gram-positive aerobic actinomyces. Okay, so there's going to be a switch in gram staining here. Don't freak out. All right, so if you're looking at like, oh, these are all gram-negatives, that's what the title says. We're actually going to switch to gram-positive toward the end. But they're fun gram-positives. They're really cool to look at, and we actually have some downstairs. So I hope to bring some of those out so you can see them too. I don't know if we'll ID them, but we'll try to look at them. Let me get ready. Everybody ready? So we start with brucella. Has anybody ever heard of brucella? So we like to start these lectures. Brucella, brucellosis. No cattle raisers in the house. Nobody's had to worry about brucellosis. Nope. Sure. It's not admitting it. It's okay. So what we have is Brucella abortus. This gets its name pretty, if you were a cattle farmer, you would know how it gets its name. It causes mama cows to abort their calves when they get infected with this. So Brucella abortus. It's also found in buffaloes. If you're like me and you're a Yellowstone watcher, right? The buffalo has made its way to the ranch, and one of the things you worry about is spreading of brucellosis to the cattle from the buffalo. So anytime you have a buffalo herd, you have to have them checked for Brucella abortus to make sure you're not going to spread it. We have one for sheep and goats and camels. It is Brucella mellitinus. Is that the way we want to say it? Or where's Cadence? How do you want to go with mellitinus? Is it good? I thought it did pretty good. I thought it was a pretty good pronunciation there. No? Pretty good?
I don't know.
Okay. What about the next one? What about the one for swine and other wild animals, a variety of wild animals? So this is the easy one, right? So if you ventured up to the northwest part of the state, you might know why they call it suey, right? You get it now? Suis. That's the way I'm gonna go with it. So I'm gonna go with Brucella suis. Suey. Let's just call it suey. That way we can put it with the pigs, the swine. And then we have Brucella canis, which hopefully everybody knows canis is for dogs. But those are our species that we have. But we're probably going to see abortus or hear abortus more, especially if you're in the animal science area and you're working on vet school or something like that. You would definitely take all of these in, but abortus is one we have to worry about with cattle farming. So the brucella specimen is blood culture. So if we suspect that one of these animals has it, definitely would take a blood culture, draw blood from them. But one of the things that you see with caution is this is one of the big worries that we have. It keeps our infection control people up at night, especially if we're doing, even on campus. When we say we're doing research on campus over in animal sciences, and when somebody has Brucella specimens that they've got out in the lab, and they've got young graduate students working with that. And if they're not trained properly, then we get accidental infections, okay? Which is your lab personnel gets infected. And 30% of all the cases are from accidental lab transfer. So when we talk about biohazard and gloves and PPE in the lab, and hey, reminder, we're actually working with Haemophilus today. You know, that's every day that we're down there. So you will have these in your lab. You'll have samples that are growing, you know, pathogens every day. And if you're, you know, not cautious and not very attentive, you can definitely get it. So we'll be under a biosafety hood with this one, because you don't want to have the aerosols, and you definitely don't want contact with skin or mucous membrane. So, you know, when we think about COVID, that's what the scenario was. First, they said it came out of a wet market from just a shopper and picked up an infected animal, right, to go home to cook, and ended up getting a jump from that to the person, and that's how it all got started. The other scenario is that most people believe is that it was lab acquired, meaning it was an infected lab personnel working with the virus in the lab, infected themselves and walked out with it, okay? So that's the scenario B versus scenario A. We may not ever know, but that's kind of what we think. Brucella species or tissue, body fluids, and bone marrow should be subcultured to blood and chocolate agar. So once we know that you have a Brucella infection, we can take any of those tissues. So if we had a cow or cat, not cat, dog, swine, goat, any of those with suspected Brucella, we could take their tissue, fluids, and things, and we could just go to blood and chocolate agar like we do for us, for a normal human micro lab. Brucella abortus requires CO2. So you need to put it in a CO2 incubator. For us, it's the candle jar. So you understand that. We can hook up CO2 gas to that incubator and have a 8 to 10% CO2 environment. Usually 6% is what we do for a hospital lab. The staining, gram-negative Coccobacillus. And we're going to see a picture here in just a second, but carbofusion is better at counter staining than what we have downstairs, which is the saffron that we do the counter stain for negatives. Again, don't forget the trick question with gram staining. What color is the gram-negative organism? Okay, after primary stain is, you know, we have to do the secondary stain. It's clear. That's the trick. They'll put that on there, and it clears. The crystal violet goes away. There is no color. It's void of color until you counter stain. So here's our gram stain. So as you know, we started picking this up when Tuesday. We started today, Thursday. We picked this up on Tuesday, which was these gram-negative rods are small, right? This group of organisms, the Haemophilus was small. You know, Brucella, that's small. So y'all had to really focus. You know, some of you are like, oh, there it is, right? Because it was so small what you were used to seeing. And it's hard to tell morphology. I mean, we can look at that slide right there and go, is that rod, Coccobacillus? I don't know. I need an electron micrograph, Mr. Director, for that. But you're going to have to call these when you get surveyed or you're investigating this. You're going to have to call that Coccobacilli gram-negative rod in the Coccobacillus shape. So it's not the whole cigar look to it. It has the diplo look to it. But that's the hardest thing. When you get gram stain surveys in, and you'll get them, and they ask you to determine a gram-negative, that's the easy part. But when they ask you, is it Diplococci, Coccobacillus, or the rod, that's the harder part. So that's very hard morphology, and it doesn't get any easier. Okay, the more you do it, of course it would, but that's a hard one. Okay. So that's when a little hint on the survey is that they give you a little background on where that sample came from. And they might say this came from a a dog source or a pig source or a cattle source. So look for those little things that give you a hint. Brucella species. These organisms are slow growers. You hold the plate for a week, okay? So you would hope that we had some idea that that's what we were searching for, you know, because who's going to say there's no growth and I'm just going to keep that plate another 4 days and see what happens, all right? So definitely you would want to have an investigative report of what we're looking for, what we think we may have. Like they may say, you know, I lost 15 calves last night. I don't know if I got brucellosis in the herd or not. Okay, that might be how that presents itself. They produce smooth colonies, but Canis, Brucella for the dog is rough, and they're non-hemolytic. So a little description of them as they're growing on the plate. Oh, there they are. It's like a blood auger. They're biochemical, they're non-motile, and they're oxidase and catalase positive, and they're non-fermenters. So if they were growing on MacConkey, they would not be picking up any lactose to turn pink. And then they would not be, they would be oxidase positive. So let's just look at that, do kind of review. So oxidase positive is usually to tell us what? Where do we use the oxidase? Well, we're on the gram-negative side, right? We're on the Coccobacilla up there in the top right left-hand corner. We see Brucella species, catalase, oxidase, and urease positive. And then then we hold the samples for 2 to 3 weeks. So we saw oxidase where? We saw it with difference between, when it came to gram-negative rods, we had a group, Pseudomonas was oxidase positive, and that's what I was hoping you'd get to, and maybe remember Pseudomonas from way back when. That was the only one that was oxidase positive that we worked with. Okay, everybody else is pretty much oxidase negative. The big ones, E. coli, Kleb, Citrobacter, Shigella, Salmonella, Yersinia, Acinetobacter baumannii. All right, so this is a summation of that. So, you know, are you going to have Brucella species to look at? You know, chances are not as much, but you never know, right? So we're covering it as our gram-negative rod Coccobacilli section. There's abortus needing CO2. The others don't need it. Hydrogen sulfide for abortus. So what is the significance? What if it did jump, right? We like to say that with the viruses, but this one jumps too. Because we think of being an infection of the cow, the sheep, the dog, the pig, but the handlers of that, or even us that ingest milk from that infected, and we know it's cows and goats, right? I don't think we get milk from pigs and dogs, right? But we do get goat milk and cow milk. And you would think, how does that happen, Mr. Director, in this day and time? You know, there are people that raise their own cows out and get milk every day. They don't go to Kroger and Walmart to buy milk. Okay, so this would be like a fresh milk, probably, because we know that what do they do to milk to make it safe? They pasteurize it, right? Louis Pasteur came up with that pasteurization, where we heat it, not to break it down, but heat it enough to get rid of these organisms. Okay. Brucellosis or undulant fever is another name for the disease. Human gets it by ingesting milk from the infected animal or just contact with the infected animal. So if you like, hang out with goats and do yoga with goats, right? Or you like to show animals at the state fair when they used to have state fairs. Doesn't that sound awful now when we say when they used to have things? Yeah, you may be, but I mean, I've seen those teenagers sleep with their cows in the pen while they're waiting on the judges to get there. I mean, they bathe them and sleep with them. You might have a pig at home, right? We all have dogs, right? Or should. Everybody needs a dog. All right, so that's how that works. So that's brucellosis. Hopefully you got an idea of the names and where they're hosting and how we end up with them in a human. So the reason the history would be very important is to know what they do. Maybe they do just come to town when they're sick, right? We do have those. Never forget, the White River produced several of our patients usually. They would live on the White River in a houseboat. And then they would come to town to the hospital. We'd say, where's your address? And they go, it's on the White River. Where? They go, I live on a boat. So you just never know where your patient may be coming in from. Bordetella. Sounds like brucella, but it's not. It's Bordetella. I think we have Bordetella on here. It's just above there. This one is in our ballpark for humans, and you've probably been infected with Bordetella and you just didn't know it. Or you did know it. We didn't do a test on you, but you presented in a way that we knew you had it. Anybody know where I'm going with Bordetella? Anybody yet? Zoom, y'all know where I'm going with Bordetella? Nope. All right, nasopharyngeal. Nasopharyngeal is where we would take the sample. We have two medias that we like to use for Bordetella. One is Regan Low, Charcot horse blood with cephalexin. And then we have Bordet. Who's somebody in here speak French? French, if I already asked that. I know Latin is somebody's, but what about French? Bordet Ginu. Would that be Ginu? Jingu? I don't think you say goo in French. I think it'd be Ginu. Potato infusion with sheep blood. But Regan Low is where I want you to remember that for sure. That's a test question, I'm pretty sure. Charcot with horse blood and cephalexin for Bordetella media. The atmosphere, it grows with or without CO2. So if it was in the CO2 incubator, yay. If it was just in the non-CO2 incubator, yay too. It would grow both places. Gram stain, gram-negative coccobacilli, we're still in that group. But we want to add 2 minutes of saffron instead of 30 seconds. And we would probably want to use carbofuchsin counterstaining instead of saffron. But we want to go longer with that to get that gram-negative. Some of you saw that Tuesday, right? Your gram-negative didn't quite look like it had any pop to it. It was kind of faded out, right? Maybe a little longer with the saffron next time. We can do fluorescent antibody stain. We make a smear from the swab. And the morphology, the little hint for morphology is mercury droplets. And I have to ask, do you know what mercury is? Do you? Where's mercury?
Like the elements?
Yeah, like the stuff. Where do we find mercury?
In thermometer?
Thermometers, yes. Y'all ever broken a thermometer open and like poured out the mercury and see what it looked like when it hit the ground? You know, I've got a story. So the story is that with Stuttgart campus, of all places, they had all of these sphygmomanometers is the way I think you call them, to do blood pressures with. They had these old ones and they got money and got some new ones, but they were like, hey, can we just bring those over to your lab and you get rid of them for us? And I'm like, why do you want me to get rid of them? They said, well, because you're the biohazard guy. That's what they call lab, you know. I said, I'm not the chemical guy. If it's mercury, but go, you bring them to camp. So this instructor brought all of these cases with these blood pressure, little manual blood pressures, right, in a box and put them in my lab. And I noticed that there was mercury running out of the box, like running. If you've ever seen mercury hit a tile floor, it runs. It beads up and it runs. It runs like a BB, but it's melted. It's like a melted BB, but it makes a BB and it runs away. And it just, it can, if you stomp it, it just goes into little bitty mercury droplets. Y'all never played with mercury?
No.
Gosh, y'all missed out. Anyway, if you get it on your skin, it absorbs in and it can actually poison you. So you don't want to ever let it hit your bare skin because it will absorb through and get your blood, okay? So all of these things are leaking mercury. And I was like, ew. So I did my best to chase it. I was chasing it down the floor with my little glass scraper, that you scrape up broken glass with. And I was, sweeping up mercury off my floor because it was leaking out of these blood pressure cuffs. So yes, that's my mercury story. Anyway, it grows, when Bordetella grows on your plate, it looks like a drop of that mercury is sitting on top. So it's kind of like a stoned colony. That's not a good picture. Let me find, oops, that's better. That's a little better. That looks like a drop of mercury. Okay. So that mercury drop, here it is, the Bordetella pertussis is the one I was hoping somebody would say, oh, I've heard of that, Bordetella pertussis. And it gives us whooping cough, right? Have y'all heard whooping cough before? No. You have? Good. Everybody gets vaccinated for it. Okay, it's part of the P of the DPT shot that you get. That's diphtheria, pertussis, and tetanus. You get that when you're little, and you get that boost up every once in a while for adults. Even to the point where some new mothers won't let you hold their babies unless you've had that current vaccination, because it's very easily spread to newborns. And if you've ever heard a newborn with a whooping cough, it's not a pleasant sound. Okay. So there's our gram-negative Coccobacilli. Here it is on Reagan low medium. That's a black media, so it's picking up. So this was one of the pictures I showed during the midterm last year, I think, and said, okay, what is that growing on that? And gave a description of a gram-negative Coccobacilli. It didn't go well. So
is it black to begin with?
yes. Yeah. So Bordetella pertussis is the clinical significant one we want to talk about. It's the whooping cough. It's basically an uncontrolled, usually at night, usually after you lay down, chronic, no production, it's dry. So you're just coughing. And if you've ever had that, you're coughing and you're coughing and you're coughing and you're coughing some more and you're coughing. And then you get up and you stand up and it kind of goes away. But if you lay back down or if the room gets hot, you start coughing and coughing and coughing and coughing. Anyway, it's just an ongoing cough kind of deal. And if you have pertussis, it give you an antibiotic, it goes away really quick. Okay, so, but if it's an ongoing cough over a long period of time is the way I like to describe it. But we do have a vaccine for it. But we are seeing more and more cases. If you get away, the anti-vaxxers are causing a little bump up in the pertussis itself. But it's respiratory aerosol, and if you're coughing, you're spreading it. Okay, so that's never good. It elaborates an exotoxin, which means that mild cough can go to severe cough, and that coughing attack can actually lead to obstruction of your airway. And it can actually lead to a death. Okay, so it's not something we just want to ignore, especially with the young ones. Okay, young babies with whooping cough are at risk. We have a parapertussis. It's a whooping cough, again, milder form. We have a bronchia septica, which is a respiratory or wound infector of Bordetella, and vaccine is the best means to protect us from the whooping cough. So up in the Coccobacillus section, we've covered Brucella, and now we've covered Bordetella. And I know those names are bang bang. They almost sound alike. They probably look alike, but they are two totally different clinical significances for us. One is brucellosis, maybe handling animals, contaminated milk from those animals, and pertussis, Bordetella pertussis is whooping cough. All right, questions so far? We doing good on time? Yeah, I think so. Y'all think we're doing good on time? Sure. Yeah. I mean, we had a delay at the beginning because we were waiting on me to do Zoom. Francisella tularenus, been around for a while. It has some Arkansas to it. That's just a good way of putting it. Okay. The state with the most infections of Francisella tularenus is Arkansas. It's also known as rabbit fever. Okay, so, but it's not an Arkansas originator. It's been around for a long, long time. It actually has its roots in Turkey. Y'all know where that is? You know where Turkey is? Like,
I didn't get over here then.
I don't know. Sometimes people like bring different animals from different places. I don't think anybody from Arkansas hunted over there to infect the rabbits over here, but it's basically from cleaning your rabbit. Okay, so it's definitely, the rabbit is the source. If you rabbit hunt and you clean your, shoot them and kill them and skin them, that's how you end up with it. So it can be breathed in or it can be contact into a wound. So it's rabbit fever or tularemia. It's highly contagious. And it was almost like, we really don't have to worry about it. Not many people are rabbit hunting these days, and the ones that are rabbit hunting know to worry about it and not get infected. But then we kind of had like a terrorist attack, and then this got listed back as a potential bioterroristic organism. So we kind of had to bring it back up on the burner to talk about. Because since it is highly contagious, it can be spread pretty easy, and you can get it from an aerosol or contact with the skin. So anytime anything's aerosolic, which you know by now, it can go through a population pretty quick, and that's why it got listed as a bioterroristic agent. It requires extended incubation, and it also, the best way to grow it is to put it on the glucose cysteine blood agar. Other media is chocolate with Isovitalix and Thayer Martin. So you're familiar with the Thayer Martin, you're familiar with the chocolate. I don't know if you're familiar with the glucose cysteine blood agar. It might be a special blood agar. Probably not going to have that one in house. The atmosphere, it grows well with or without CO2, and they're small gram-negative rods. So Francisella tularenus. There it is growing on the, that looks like the cysteine, what was it again? Cysteine glucose blood agar. That looks like chocolate. And no, there's the glucose cysteine media, so that just must be, I don't know if that's blood agar or not. It also grows here on one that we see with buffered charcoal yeast extract. That's maybe, have we had that one already? We've had that one talked about, BCYE agar. Okay. We're really not going to search out for it. ID should only be done in a reference lab. So this would be one of those if you suspect a bioterroristic attack. I did this years ago with the health department. We had to go through a training on And if you go into the old, if you go into the micro labs of any hospital, you'll see this big poster from the health department. And it's got several of these organisms, pictures growing on them in case you see this. This is what it looks like, and then you suspect that it could be a bioterroristic attack, and you're supposed to gather up all your cultures and seal them legally and send them to the health department for ID. So that's what this means. ID should be only done in a reference lab or health department lab. So FYI, oxidase negative. We see it up here with our Coccobacilla. It says requires cysteine and iron for growth, tularemia, ticks and rabbits, and BCYE agar. So you have a little helpful hint on your reference sheet. The disease it causes is lymphadenitis, pulmonary tularemia. That's the one we worry most about with the bioterroristic attack. Ocular granular tularemia gets in your eyes because you rub your eyes. Transmission is the tick bite causes the lymphadenitis, biting through the skin. The pulmonary would be, hey, I'm skinning my rabbit and I inhaled an aerosol that was infected from the pelt of the rabbit with tularemia, and I got inoculated that way. And then ocoglandular is wiping your eyes after you cleaned your rabbit. So you can rub your eyes like this, and all of a sudden you've got eye infection with tularemia. So there's our dermacenter tick. And this is what it would look like. You got a, on your hands, see that, see that. Here, we didn't, I mean, around the eye, I guess, but on the wound of the hand. All right. So that one gets classified as the Arkansas rabbit fever organism because about 35% of all cases in the United States are in Arkansas per year. There's a certain group of states, about half of all the, infections occur. That leads us to Pasteurella multocida. Do we have Pasteurella on here? I think we do, don't we? Is it still up in that? Oh yeah, it's still up in that Coccobacillus section. This one is our cat and dog bite. Okay, so Pasteurella multocida. I know I have, if you can remember this from now, I will tell you there is a case study that will appear sometime if you're lucky enough. I don't know which class, I can't remember if it's clinical or if it's micro 2, but I'm pretty sure it has a bite on the hand of a cat. Okay, so come back to Pasteurella multocida when you get there. Hopefully you'll remember that. It is a gram-negative Coccobacilli, wounds, blood, respiratory specimen from the human. No special media required, grows well on blood and chocolate agar. And just so you know, chocolate agar is pretty much there for every wound culture we do. So anytime we've got a wound or an abscess or something that sounds wound type, then we do put chocolate agar in the mix. Here it is, Coccobacilli, gram-negative. There's no way I could tell you from this one or the other one that we had, the gram-negative. I can tell you it's gram-negative. I can tell you it's Coccobacilli, and I can tell you what group of organisms we may be looking at. But am I going to be able to tell you that's what it is off the gram stain? No. Or the blood agar. The one thing that we associate with Pasteurella multocida is that we talk about coins. So it's a mnemonic. Is that what you call it? Not a mnemonic, just a brief. What is the mnemonic? It would be the sounds, but coins are the first letter of all the other words. What is that called? Somebody knows. Zoom, do you know? Anybody know when you take the first letter of something, it spells something out?
I do not know what you're talking about.
It's a study. It's like a study guide.
I don't know what it's called.
There's got to be some name to it. Gosh.
Because there's one, this is Randy, there's one for math remembering like PIMDOS. It's
That's
the
too
same
much
thing,
for me.
but, like,
Yeah,
I don't know what it's called.
I get the ones like
Are you sure
green
it's not
green
that?
boy or two boys fighting. You know those kind. Y'all know those? Yes.
Is it acronym?
Acronym. That sounds good. Thank you.
I'm looking.
Thank you for looking that up. Yeah, somebody's got Wi-Fi in here, right? Biochemical identifier, catalase, oxidase, indole, nitrate, sucrose. So that'll help you knowing Pasteurella positive for all the coins. If you can remember C's for catalase and O's for ox, you should. We've done that enough. Indole, we've done that. Who else is indole positive? That's a gram negative. See how well you are keeping up with me. Who else is indole positive as a gram negative rod? Be automatic.
E. coli.
E. coli, thank you. There we go. All right, nitrate positive, sucrose positive, coins associated with Pasteurella multiseta. Clinical significance, cat and dogs. Upper respiratory tract, bite. Cat bite, dog bite, cellulitis follows, abscess follows. Yes, the dog bit me and look, I've got this big abscess coming back. I've got cat bite. What's the significance there? And then all of a sudden you get this cellulitis. So definitely associate Pasteurella multiseta with cat and dog bites. All right, that's the gram negative story. Told you we were going to switch gears. We'll switch gears to the aerobic actinomycetes. Actinomycetes. Yeah, I'm going to go with CDs because I got to remember I got the ES at the end. But this is a gram positive. Yay. So now we're introducing our gram positive rod right here. So we've got another lecture right behind this next Tuesday with more gram positive rods, but we're going to go ahead and make the switch here since I guess we have a few minutes to do that. So if you found it on your sheet, you flipped it over and found it. I think I got one over here, Actinomyces israeli, or Israeli. Probably located in Israel, if I was guessing, but we don't know for sure yet. It's aerobic, branching filaments, and gram-positive rods. So these are cool looking. And this was on Facebook just the other day. One of my micro friends posted a picture of it from his sample. He had one. It was a Nocardia species. You cannot confuse it, but you can. You can confuse it with mycology. So don't make that mistake. These are still bacteria, okay? But when you see these branching filaments, you're going to think, that looks like a yeast growing over there. But Nocardia stains unevenly with the gram stain, therefore it exhibits a beaded appearance. So that was the description that was on Facebook from my friend Nick up in Chicago. He said Nocardia stained, I got a beaded stain, gram positive, what do you think it is? And Nocardia was the go-to answer. And one of the things you need to know is partially acid fast. So probably going to get this from a sputum, okay? And anytime we get a sputum, we do acid fast for Mycobacterium. And we hadn't gotten there yet, but we're getting there. But there it is. And if you saw that in about 1st of November, when we get into mycology, you're going to say, oh, those are hyphae, and that's a mycology. That's a growing fungus there, right? Yes, you would probably do that, but this is Nocardia, okay, doing the same kind of thing, but it's a gram positive rod. So they just, oh, you just want to go, that's mycology, that's hyphae, that's branching hyphae. I need to get in there and determine what it is. Oh, yes. So here's the partial acid fast. Acid fast, we do that looking for red. So that's why it's called partial, because it's kind of halfway between purple and red. Here's the gram stain to the right of the Nocardia species. So we put the two together. We have aerobic actinomyces. We have Nocardia and Streptomyces. So we have two. Those are the aerobic. We have anaerobics. Okay, the anaerobics are actinomyces species, and we're going to see the anaerobics later. We're going to come back for all anaerobes, and I think about 14 and 15 lectures. About the 20th and 22nd of October, we'll have the gram negatives. I mean the anaerobics. Sorry. The habitat, these are found in the soil, decaying plant matter. So where you're going to pick up Nocardia and Streptomyces. Nocardia may colonize the skin or the upper respiratory tract. So you may have, or you may have some kind of injury due to implantation. And we think of that with thorns. We think of stickers, right? Getting stuck as you walk through barefooted into something, or your hand, you're gardening and you're getting all the brush up, and all of a sudden you look at your hands, you've gotten stuck like 3 or 4 times with something sharp inside that group of limbs that you're dealing with. Those are kind of the traumatic injuries, especially immunocompromised. So we can have abscesses and tissues, but sputum is usually where we'll pick up for this respiratory nocardia. We use a gram stain for smears. We also use GMS stain, and that's Gamori, methenamine silver, methamine silver stain. We also have hematoxylin and eosin, H and E stain, and you'll see that if you've ever been in histology lab working at all. When they do tissue sections, they do H and E stains a lot on tissue. If you're wondering where that, you may see that. If you had, some labs have histo attached, cyto and histo, and you'll be in there with cytotechs and histotechs. And it's a different degree, but they can definitely be connected to your lab. And especially if you have pathology specimens, that's where the histotech and cytotech are. They're looking at cells and they're doing tissue sections from malignancy, cancer cells, and that type do. They're prepping it. Thin prep. It's really neat. You ever seen a hematoma or hematome that does tissue section? Like when you get something removed from your body and they freeze it and they send it off to be analyzed, they'll take that like a block of tissue and they'll make little thin slices and then put it on a slide and stain it for the pathologist to look at to grade out if you've got bad cells or good cells. Or y'all never heard of that? Y'all heard of histotech before? No? Histology at all? Cytology at all?
This degree to do that?
You can definitely go to those. Yeah, we have some. I have Little Rock has a cytotech program, and the MLS have gone into that as kind of like a subspecialty. Use their knowledge of what we teach and then take it to the next level of a different, little different angle. Kind of makes you a little more marketable with cytotech. It sells big. I mean, they pump it up a little bit down there. They say the jobs are very, very hard to fill. They're looking for cytotechs and they're very high paying. So if we get to go to if we actually get to go to Little Rock in the spring for Quiz Bowl, I'll introduce you to the cytotech program director. She'll be there at the conference. So yes, that's some subspecialties, but yeah, definitely could go double up. You can't, I don't think you can do both. You have to choose one. There'll be some students, that chose cyto first, could always come into lab second, and then some that did lab first and go to cyto second.
And it pays more than the bachelor degree here.
They say it does, but I think they're moving it to a master's level. So that's key because that's the difference is you would get to do a second level degree in cyto with a master's attached. And that would be very helpful. So it's definitely something to think about. If it's that, I think they moved it to masters, so I think they're in the process of doing that if they hadn't already done it. And that's always key. You don't want to do a second bachelor's. You know, you want to try to get the masters out of the way. That way you can teach if you want to. All right. Respiratory specimens. We're going to concentrate them with acid-fast as with acid-fast bacilli. So that's why I said these are kind of handled the same way. Like if you were looking for Mycobacterium, TB patient with a sputum culture, we do the acid-fast instead of waiting the 6 weeks for it to grow. Cerebral spinal fluid, definitely could see nocardia there, or not, yeah, nocardia in urines. Swabs are not recommended because they just don't get enough of the stuff. So we definitely want like a sputum culture or a body fluid. Since it is similar to Mycobacterium, we do the 7H10, or we can use brain heart infusion agar, as well as Lowenstein-Jensen slants. So a couple of new names. I know we have brain heart infusion. Y'all did that Tuesday. You use that media for your VX strips. We just used a nutrient agar and didn't do any blood in it. I don't have any 7H10. And the thing here, it says, caution, do not use media containing chloramphenicol. Yeah, that's a tough one, because I think that's all your notes say, right? Don't use chloramphenicol in the media. Anybody know why we wouldn't use chloramphenicol in the media for this organism? See who's going down the right thinking train and surprise me with this answer.
Is it because it's chlorophyll based?
Chlorophyll based? Simpler than that. Simpler. Think about treatment. What do we treat the infection with? Chloramphenicol. So why wouldn't we want to put that in the media with it?
It's not gonna grow.
Not going to grow, right? Not going to grow. So we don't want to have that in there if that's what we treat it with, because it's going to prevent growth. Temperature 35 degrees Celsius, which is a little cooler, and CO2. Y'all see a board question on this? Can y'all pick out board questions yet? I think I just saw one.
Should we be able to?
Eventually you will. A board question. That last sentence it looks like a board question. With nocardia and Lowenstein-Jensen slant, would you incubate at 37 or room 10? That just screams board question right there for me. Anyway, may never see it again. Here's Lowenstein-Jensen media with nocardia asteroids on there, asteroides, and it doesn't look like anything we've seen before. It looks like a fungus growing or a yeast growing. We'll see a bunch of this in November toward December as we finish up the semester. That one doesn't do it justice with the asteroides on 7H10. It's growing there, but that's the classic. That is the as I would say, that is the. My daughter has these in her room. These little, what are those? Those little plants. Succulents. There you go. No, somebody knew that. It kind of looks like a succulent to me. I don't know if it does to y'all, but that's what I think when I see this. I think succulent. I can see this growing in my daughter's room, and she's like, "Don't spray water." No. Anyway, it's unique for sure.
So are we gonna grow this or not?
No. Well, I say that. I think I have it. I was looking. I know I don't have tularemia because I was looking for that. I was like, I hope I don't have that. Double check it to make sure. In case I did, I'd grow it. Even though it's a biohazard. I mean, a bioterroristic organism. But I think I have this. So we'll look at this. So we may grow this out after the midterm's over. The thing we got to do is we incubate it for 4 weeks, though. So nocardia species grow as a pellicle thin film along the surface, granules at the bottom of the tube, and on a blood auger, they're chalky white colonies, which you just saw. So there they are. Our mycetomas are what infection would cause. It could be caused by a fungi. It's called a eumycotic mycetoma. But guess what? Actinomycetes, the gram-positive rod, causes the mycetomas too. So this is not only a fungal, that's why I'm saying this is kind of, is it a fungus or is it a gram-positive rod? The mycetoma is an infection involving bone, cutaneous or subcutaneous tissue. We have swelling, tumefaction or drainage in the sinuses or granules. Granulomatous tumors develop in the subcutaneous tissue, and the port of entry is a traumatic implantation of a thorn or a splinter with the nocardia. So this would be, if you've ever experienced, going through the woods and all of a sudden you impaled yourself on a limb that had these thorns on it. Some trees have thorns on them. Well, these could be present in those and definitely introduce them to your subcutaneous tissue. Nocardiosis is what we call the infection, localized disseminated disease. Originates in the lungs after you inhale the organism, and it resembles tuberculosis. So you would be that determined, right? So that sputum culture that comes out as nocardia, which was a partial acid fast instead of a true acid fast. We've got some differences between Mycobacterium and nocardia, but it will come from kind of the same place if it's a respiratory. The Astroides is the most common cause of nocardiosis. We have Braziliensis in Mexico is the other nocardia. Again, looks like a board question to me when we're sitting here focused on Astroides and all of a sudden we bring in Braziliensis with Mexico. Clinical presentations of nocardiosis is inflammatory lesion with pus and necrosis. We can stain the tissue section. We look for gram-positive branching filaments, which we have seen already, rod-like bacilli or coccobacilli, fragmentation, and it looks like that's behind this picture if you're looking at the slideshow. And then for biochemical identification, we have nocardia, Astroides down here. This is calcium sulfide, I think calcium sulfide, the gelatin again, hyperpuric acid, tyrosine, xanthine, urease, lysine. So we have our players in here, but would you go to that? I mean, you're not going to miss it. I mean, if you saw it growing, it would be pretty obvious. If you saw it on a stain, it'd be pretty obvious. But you do have some biochemical identifiers that you can use to differentiate the different species.
Is calcium sulfide the same as casein?
Case.
Yeah. That's what it says on our paper.
What does it say?
Casein?
Casein? Yeah.
Yes.
Well, maybe, I could be case. I don't know. I have to double check. I thought it was calcium sulfide when I saw CAS, so I'm sorry. It says casein on your thing.
Yeah.
Yeah.
I was just gonna mark it if it was the same thing. I didn't know.
Go with casein then.
Okay.
I didn't look at that, sorry. I looked at calcium sulfide for CAS. But I'll go with casein.
I wasn't
Alright.
trying to correct you. I was just asking.
No, I didn't notice. So
You know.
definitely, yeah, I don't mind that at all. Don't ever think you can't, like, hey, is that, you just said calcium sulfide, but I got casein written down. Because I see abbreviations, you never know. Caution, due to graphic, we finish up with some pictures of nocardia and the subcutaneous. tissue, an impalement. So you could see that was probably some kind of impalement into the arm that led to this. That just, yeah, that looks like morula or something. That's ugly back there. All right, that's it. All right, so we'll stop the share. Y'all got any questions? All right, we're gonna stop the recording.